Microscopy. Understanding how we study cells. Microscopy is the science of studying extremely small objects that cannot be seen with the naked eye. In biology, it is mainly used to observe cells and the tiny structures inside them. Cells are incredibly small. Because of this, scientists use microscopes to study their shape, structure, and internal parts. Over time, as technology improved, microscopes became more powerful and allowed scientists to see more detail inside cells. Today, the two most common types of microscopes used in biology are light microscopes, electron microscopes. Each type has different strengths and is used for different purposes. Light microscopes. A light microscope works by using visible light and a system of glass lenses to produce a magnified image of a specimen. When light passes through a thin sample, the lenses bend the light and enlarge the image so we can observe it through the eyepiece. Light microscopes allow us to see individual cells, large structures inside cells such as the nucleus, basic cell shapes and arrangements. However, light microscopes have a limit to how much detail they can show. Very tiny structures inside cells cannot always be seen clearly using light alone. Electron microscopes. Electron microscopes work in a different way. Instead of using light, they use beams of electrons to create an image. Electrons have much shorter wavelengths than light, which means electron microscopes can produce images with far greater detail. Electron microscopes have two major advantages. First, they provide much higher magnification. This means the image can be enlarged far more than with a light microscope. Second, they have higher resolution. Resolution refers to the ability to distinguish two points that are very close together. The higher the resolution, the sharper and clearer the image. Because of this, electron microscopes allow scientists to observe very tiny structures inside cells, including the internal structure of mitochondria, the structure of chloroplasts, ribosomes, plasmids. These structures are usually too small to be seen clearly with a light microscope. The development of electron microscopes has helped biologists greatly improve their understanding of cell structure and how cells function. Using a light microscope required practical. In GCSE Biology, students must understand how to properly use a light microscope to observe cells. This includes preparing a slide, focusing the microscope, observing the specimen, drawing and labeling what is seen, preparing a microscope slide. Before observing a specimen under a light microscope, it must be placed on a microscope slide. A microscope slide is a thin piece of transparent glass or plastic that holds the specimen. For example, if we want to observe onion cells, we prepare the slide using the following steps. First, place a drop of water in the center of a clean slide. Next, cut a small piece of onion and separate one of its layers. Using tweezers, carefully peel off a very thin layer of epidermal tissue from the inner surface. Then place this thin tissue sample into the drop of water on the slide. After this, add a drop of iodine solution. Iodine is a type of stain. Stains are chemicals that add color to parts of a cell so that they can be seen more clearly under the microscope. Some cell structures are almost transparent, which means light passes straight through them and they are difficult to see. Stains solve this problem by highlighting different parts of the cell. For example, iodine is commonly used to highlight starch in plant cells. Eosin can make the cytoplasm easier to see. Finally, place a cover slip over the sample. A cover slip is a very thin square of glass or plastic placed on top of the specimen. It helps flatten the sample and prevents it from moving. When placing the cover slip, it should be lowered slowly at an angle to avoid trapping air bubbles. Air bubbles can interfere with the image and make observation difficult. Observing the specimen. Once the slide is prepared, it can be placed on the microscope stage. The stage holds the slide in position while it is being observed. To use the microscope correctly, follow these steps. First, secure the slide on the stage using the clips. Next, select the lowest power objective lens. This produces the lowest magnification and makes it easier to find the specimen. Then use the coarse adjustment knob to move the stage upward until the slide is just below the objective lens. After that, look through the eyepiece and slowly move the stage downward using the coarse adjustment knob until the image begins to come into focus. Finally, use the fine adjustment knob to sharpen the image and make it clear. If greater magnification is needed, the objective lens can be switched to a higher power lens. After changing lenses, the focus should be adjusted again. Drawing what you observe. In biology, drawing scientific observations is an important skill. When drawing cells viewed under a microscope, several rules must be followed. The drawing should be made with a sharp pencil and clear, continuous lines. Shading and coloring should not be used. The proportions of the structures should be accurate, meaning the sizes of the parts should reflect what was actually observed. The drawing should include a clear title describing what is being observed. Important structures such as the nucleus, cytoplasm, chloroplasts, or cell wall should be labelled using straight lines that do not cross each other. Students must also calculate and include the magnification of their drawing. Magnification. Magnification describes how much larger an image appears compared to the real object. It is calculated using the formula. Magnification equals image size divided by real size. For example, if the image of a cell measures 2 millimetres and the real cell is 0.02 millimetres wide, the magnification would be 2 divided by 0.02 equals 100. So the image is magnified 100 times, written as 100. Calculating image size. Sometimes the magnification and real size are known, and the image size must be calculated. The formula becomes image size equals magnification real size. For example, if a cell is 0.3 millimetres wide and the microscope magnification is 50, image size equals 50.3. Image size equals 15 millimetres. Calculating real size. If the magnified image and magnification are known, the real size can be calculated using real size equals image size divided by magnification. For example, image size equals 10 millimeters. Magnification equals 400. Real size equals 10 divided by 400. Real size equals 0.025 millimeters. Standard form. Because cells are extremely small, their sizes are often written in standard form. For example, 0.025 millimeters can be written as 2.5 10 to the power of 2 mm. Standard form makes it easier to express very small numbers clearly. Units used in microscopy. 3 common units are used when measuring cells: millimetre, micrometre, nanometre. Their relationships are 1 millimetre equals 1,000 micrometres. 1 micrometre equals 1,000 nm. When calculating magnification, it is important that both the image size and the real size use the same units. For example, if the image is measured in millimeters and the real size is in micromicrons, one of them must be converted before calculating. Estimating the size of cell structures. Sometimes it is difficult to measure the exact size of structures inside a cell. In these cases, scientists estimate the size by comparing it to the size of the whole cell. For example, if a cell is 60 micrometers wide and the nucleus appears to fit across the cell about 8 times, the approximate width of the nucleus can be calculated. Width of nucleus equals 60 divided by 8. Width of nucleus 7.5 micrometers. This method allows scientists to estimate sizes when precise measurements are not available. Estimating area. The area of small structures can also be estimated by comparing their shape to a simple geometric shape. For example, a mitochondrion often looks similar to a rectangle. The area of a rectangle is calculated using area equals length, width. If a mitochondrion measures, length equals 4.6 micrometers. Width equals 1.5 micrometers. Then the estimated area would be area equals 4.6 1.5. Area 6.9 square micrometers.
Important exam skills.
For GCSE Biology, students must be able to explain the difference between light microscopes and electron microscopes, understand magnification and resolution, prepare and observe slides using a light microscope, draw and label biological structures accurately, use magnification formulas, convert between units such as mm, μm, and nm. Estimate the size and area of cell structures. All of these skills are commonly tested in GCSE biology exams.